Good afternoon, dear students. The Department of Histology represents for you the lecture Cartilage and Bone, Skeletal Connective Tissue. The skeletal tissue can be classified for cartilage and bone, like a classical skeletal tissue, and specialized tissue of teeth, dentine, and cementum. All these tissues originate from mesenchyme, but different sources of mesenchyme, because we have known about the mesenchyme like a precursor tissue rudiments that's to originate, to give rise to the formation of all type of connective tissue, vessels, and of course skeletal tissues also. So we have seen here the hyaline cartilage, elastic cartilage, fibroelastic cartilage, and compact bone-like examples of this type of tissues. So first question about the cartilage. Cartilage is a durable form of supporting connective tissue characterized by an extracellular matrix with a higher concentration of glycosaminoglycans, GAGs, and proteoglycans, interacting with the collagen and elastic fibers. Structural features of this matrix make cartilage ideal for the variety of mechanical and protective roles with the adult skeleton and elsevier. Cartilage consists of cells called chondrocytes, chondros from cartilage, and cytos cells. These cells, chondrocytes, embedded in the extracellular matrix, which, unlike connective tissue proper, contains no other cell types. Chondrocytes synthesize and maintain all extracellular matrix components and are located in the matrix cavities and called lacuna. Look at this one. We have the components of extracellular matrix, which is very visible isogenic groups, the specialized structural elements, proteoglycans, and glycosaminoglycans, which is present here like a green color elements. Largest molecule of proteoglycans is, glycosaminoglycans is named hyaluronic acid. This is green color core. It's a structural base or core for these molecules. And from this core, we have seen some junction proteins, and it's connected with the specialized proteoglycans and sulfatic glycosaminoglycans. And this is in cartilage, we're talking about chondro-18 sulfate. Chondro-18 sulfate. That's molecule glycosaminoglycans and proteoglycans characterize higher ability to communicate, interacts with the water molecule. And because that we have talking about the free water molecule and junction joint water, which is interacts and joined with this molecule. So we have the specialized diffuse effect of water from the perichondrium into the extracellular matrix of cartilage. Because that inside cartilage proper absence any vessels, and this is isogenic group we have seen here. Two chondrocytes joined together, and each chondrocytes housing in the special lacuna, and this is the area ordinarily filled by the specialized extracellular matrix, which is named chondromucoid. Such as between this is lacuna, between this is isogenic groups, we have seen the intraterritorial matrix. So we have talking about territorial matrix inside the isogenic group and intraterritorial matrix between. Some of the materials which interacts with this matrix will be fibrous component, fabric component. Fabric component in the cartilage, ordinarily this is collagen type 2, sometimes collagen type 1, and elastic fibers. So this is elastic and collagen type fibers is a product of chondrocytes. Chondrocytes effectively to synthesize proteins, collagen and elastin in some type of cartilage, and to support the quantities of the fabric and amorphic components, a ground substance during the functional conditions. So the role of chondrocytes to support and synthesize the extracellular matrix. By the way, chondroblast, specialized cells outside the cartilage, ordinarily they are located in the perichondrium, in special layer, surround the cartilage, connective tissue, ordinary dense connective tissue. This is cells chondroblast generate a new population, new lineage of chondrocytes. Next schematic diagram to show for us the development of skeletal tissue, and we have seen that the cartilage and bone originate from mesenchyme. But the mesenchyme can be classified like two types of mesenchyme. The first type of mesenchyme, this is mesenchyme which originate from sclerotomes. This green color elements is a sclerotome. Sclerotome of dorsal paraxial mesoderm, somatic mesoderm. Is, if you remember, surround the neural tube, we have the condensation of mesoderm, and this is mesoderm is named dorsal paraxial somatic mesoderm or mesoderm of somites, and each somites composed from dermatomes, myotomes, and sclerotomes. This green color elements, this is green color area, is named sclerotomes. So sclerotomes give rise the formation of skeletogenic mesenchyme. Skeletogenic mesenchyme, which to forming the skeleton. Primary, it will be hyaline cartilage, like a modeling of the future skeleton. After time, it will be replacement by the constant compact bone. As another area of the head, like example in the skull, we have talking about the ectomesenchyme. Ectomesenchyme occupied the area of skull, and ectomesenchyme originate from neural crest. As you remember, neural crest is extra dosage of neural ectoderm, which is migrates after the closing of neural tube as a result of neurulation. The quick picture to show for us the source of this one is neural crest cells, which is migrated after neurulation, and this is red color somite, and the portion near the neural tube is named sclerotomes. Sclerotomes to give us a formation of cartilage and bone everywhere except then in skull. Development of skeletal tissue characterize the specialized process, like example, aggregation and condensation of mesenchyme. This is aggregation and condensation of mesenchyme to take an active part in the formation of the future limbs and axial vertebrum as a result of controlling of special Hox genes. This is Hox genes, is a name like Hox, yes, short name, to remodulate or modulate the structure of a skeleton via the producing the special signal molecules. The famous signal molecules, which is named bony morphogenic proteins, like famous is BMP2 and BMP5, and also endogenic regulators like growth, transforming growth factor beta, and glial differentiation factor GDF5. So this is factors GDF5, bony morphogenic proteins, and transforming growth factor beta to active participate in the regulation of differentiation mesenchyme into the skeletal tissues. What about exogenic factors? So the endogenic we discussed a minute ago, it was being Hox genes and special signaling molecule like growth factors. But now we are talking about exogenic factors. Main of them is oxygenation. In the condition of hypoxia, the loss of oxygenum, the stem cells of skeletogenic mesenchyme straight differentiated into the chondrogenic stem cells. From another condition, after 8 weeks, in the 9 week and late, start the normal oxygenation, and this is oxygenation normal as a result of completing of placentation, and through the placenta starts the active migrate the oxygenum, and this is oxygenum to play the key role in the stimulation, initiation of formation of osteogenic stem cells. So, what's the main idea? That's in hypoxia, we talking about modeling the skeleton like a chondrogenic models. And after that, all long bones, vertebrum, it's originate from chondrogenic models through the chondrogenic stem cells. After time, replacement by the osteogenic transformation via endochordal ossification. But around the 8-9 week of development, we're talking about the straight formation of osteogenic stem cells. In this case, we're talking about intramembranous bone formation. Intramembranous bone formation. What about the hondogenesis? We start first of all discuss hondogenesis, the formation of cartilage, formation of cartilage. Primary formation of cartilage, it starts in the 5th week of prenatal development. We have identified stages of development. Formation of chondrogenic islets. This is a key moment of the beginning of the formation. The main should be play key role epithelia-mesenchyme interactions. So interactions between the mesenchymic islets, primary aggregation, and epithelial lining of surface ectoderm will to give rise of formation of epidermis and some other, maybe some glands. And in this moment, these elements of ectoderm, epithelial lining, to stimulate the regional transformation of the area of future joint into the mesenchyme, so mesenchyme transformation into the cartilage. The stages of this process: procondensation of mesenchyme, first stage, Next, condensation of mesenchyme, and the next is chondrogenic differentiation. How does it originally take place? We have seen in this schematic diagram. So look at this one, stages of development. First stage, this is a pre-condensation, when epithelial mesenchymal interactions between mesenchymic cells and epithelial lining to activate from the MSX1 gene and BMP factor 2 bony morphogenic protein transforming growth factor beta 1 and tenascin. This is specialized proteins activate the process of epithelial mesenchyme mesenchymal interaction. Effect of it, this is mesenchyme start to condense, precondensation and condensation. During the condensation of mesenchyme into the chondrogenic islets, start the new active molecule communicate, interact each to other, and to results of this interaction, we're talking about the chondrogenic islets formation. First of all, it will be transforming growth factor like signaling molecule, fibronectin like a mediator, activin like activator, and NCAM protein, which to result we also have seen that syndican we can use also in the in vitro techniques. Yes, and all this process is controlling by the Hox genes MSX1 and 2 and bony morphogenic proteins 2, 4, and 5. Results the formation of mesenchymic islets. And the next after this interaction is a result of the proteins activations, the mesenchymic islets transform into the cartilagenic islets or special cartilagenic plates in any area. What about the hondogenesis? More effective description. So primary hondogenesis, tissue formation, that's a prechondroblast. This is cells which is a primary appear from mesenchyme, prechondroblast. Next stage, this is chondroblast. Next, prechondrocyte. So this is a differon of chondrocyte cells, and the next cells will be chondrocytes as like differentiation, which is produced extracellular matrix. Classical variant of the extracellular matrix in cartilage is named chondromucoid. Anywhere area is named chondrine globe. So formation of perichondrium to take place in the bordering of chondrogenic islets to aggregate cells from the mesenchyme that transform it into the dense connective tissue and vessels at the periphery of each islet. There are two mechanisms of cartilage growth exist. The first type of cartilage growth mechanism, look at this red link, yes, we have oppositional growth that provides the perichondrium forming a new layer of the matrix at the periphery of existing cartilage, ordinarily via the synthesis of collagen type 1 and 3, of course by the chondrocytes activities, and interstitial growth in the process that forms the new cartilage within the cartilage mass by chondrocytes divisions. So here chondroblast, chondrocytes, here only chondrocytes. So interstitial growth is the process that forms the new cartilage within the cartilage mass by the chondrocytes divisions, forming the isogenic groups and intracellular matrix secretion. Ordinary in the second growth, interstitial growth, take place the synthesis of collagen type 2. Effectively, it's happened in embryonic time, but very often it's also used during regeneration after injury. What about the maturation? What about the differentiation of cartilage? As you have seen, this is schematic diagram from the book of Jangueira, Basic Histology. We have seen the major stages of embryonic cartilage formation or hondogenesis are shown here. The mesenchyme, litera A, mesenchyme is a precursor for the all type of cartilage. You have seen the stellate cells, which is like resemble on the like stars or maybe spiders, and this is cells have very active proliferate. This is mitotic figure is visible, like to symbolize that this is very active dividing cells. And mitosis and initial cells differentiation produces the tissue with the condensation of rounded cells called chondroblast. And the next will be the chondroblast cells. We have seen that the chondroblast cells also mitotically active and chondroblast to generate a new layer of cartilage. Chondroblasts are then separate from one another again by the production of the various matrix components, which is collective swell with the water and form the very extensive extracellular matrix. As you have seen here in the letter C, we have how the extracellular matrix chondrine globe is appears around each of chondroblast, yes. But the multiplication of chondroblast with the matrix gives rise to isogenic group, isogenic cells formation. That is, aggregate surrounded by the condensation of territorial matrix. So the territorial matrix, this is a site of the extracellular matrix, surround the cells inside isogenic group, territorial matrix. In mature cartilage, these interstitial mitotic activities cases and all chondrocytes typically become more widely separated by their production to the matrix, and surrounded is visible extraterritorial or interterritorial matrix. Here is very good seen the special chondrogenic islets and future chondrogenic plates. So what about the layers of the formation of the matrix and the layer of cartilage? First of all, at the periphery, in this schematic representation, all those abundant molecules of cartilage matrix shown the interactions between type 2 collagen fibers and proteoglycans, green color, linked to the hyaluronan or hyaluronic acid. Link proteins, non-validative blind the proteins core proteoglycans to the linear hyaluronan molecules. The 118 sulfate side chain of the proteoglycans electrostatically bind the collagen fibers to form the cross-link matrix. Is very good visible. How does it happen? And we have talking about the cycle area is shown a larger and lower part of the figure. Link proteins, core proteins, 118 sulfate, collagen type 2. Physical properties of these matrix components produce a higher hydrated pi-level material with a great strength Approximately 75% of weight of the hyaline cartilage is water. In the diagram number B, we have seen the transitional area between the perichondrium and the cartilage matrix. This is a perichondrium, this is a cartilage matrix. Ordinary chondroblasts occupy the perichondrium, but the lowest portion of this is this is original cartilage. And we have seen interterritorial matrix, chondrocyte, territorial matrix, interterritorial and territorial matrix. Of course, this matrix is a different condition. So, these function cells produce the matrix component and exists the lacuna surrounded by the matrix. The extracellular matrix immediately around each lacuna, called the territorial, remember, contains mostly proteoglycans and spares collagen that more distant from the lacuna. The interterritorial matrix is richer in collagen and may be less basophilic. So we have talking about to have visible differences in chemical conditions of this area. Is more basophilic territorial matrix and less basophilic interterritorial matrix. Okay, and we have seen surrounding connective tissue, perichondrial fibrous layer, chondroblast layer, and the layer of maturated and mature cartilage. The cells of cartilage, chondroblasts, secrete a large amount of proteins and carbohydrates to produce the intercellular matrix. Therefore, they have an abundant rough endoplasmic and smooth endoplasmic reticulum, the Golgi complex, and numerous mitochondria. Chondrocytes are older cells embedded in the intracellular matrix. They cannot secrete matrix anymore and contain a few organelles. So this is the differences between the cells. Look at this one. We have seen chondrocytes. This is also chondrocytes. And we just remember this is picture of examination, electron transmissional electron micrograph. In isogenous group of chondrocytes is very good visible nucleus number 1, cytoplasm is rough endoplasmic reticulum, intercellular substances of cartilage, amorph cartilage under the number 3. This is this is a territorial matrix, and collagenous fibers on the bordering between territorial and interterritorial matrix. Also, we have seen here same another photo of the cells. We have seen territorial matrix and interterritorial matrix surrounded. Also, very good visible rough endoplasmic reticulum and some lysosomes to transform the surrounding matrix to participate in the activities of remodulating of the matrix structure. And we have seen how the cells interact each to other with the processes. Also, we use a special metachromasia for identifying the area of territorial matrix. Look at this one. It's very good visible how to reagate the hyaline cartilage for the different type of stain, like example, the stain toluidine blue with alcyan. And we have seen the differentiation of the type of the matrix. The territorial matrix is more pink, and the interterritorial matrix here is visible more basophilic, but in here is interterritorial less basophilic, more oxyphilic. As we was discussed a minute ago, basophilia, it's a characteristic of territorial matrix, and interterritorial is ordinarily less basophilic because more oxyphilic with you have seen here. Next moment, the cartilaginous matrix. It's a firm but still pliable, uh, pliable, still pliable. Collagen fibers are prevalent in constitute about 40% of the total matrix, but they are not visible in histological section because the fibrils are very fine and have the same refractive index as the ground substance. The amorphous ground substance enriched in proteoglycans, hyaluronic acid, hyaluronan, chondroitin sulfates, and keratan sulfate. The matrix adjacent to the chondrocytes, the capsular matrix, is poor in collagen but rich in glycosaminoglycans and stains deeply basophilic. The cartilage matrix is highly hydrated. Much of this water is tightly bound to the proteoglycans aggregates, which explains the turgor of cartilage and higher matrix permeability for the diffusion of oxygen and nourishment from the blood. And I want to tell you, so very important to understand that look, the cartilage lacks blood vessels. Then if we have compared the cartilage with connective tissue, in the cartilage, blood vessels not present in cartilage proper area. It's located only in the perichondrium. Nutrients, waste materials, and hormones pass between the blood vessels and cells via diffusion, except higher molecular weight proteins like immunoglobulins. The cartilage transplantation in humans is successful. So we can use the transplantation of cartilage very easy because the vessels is not present inside the cartilage. There are the three types of cartilage: hyaline, elastic, and fibrocartilage exist. Look at this one. We have seen the position of this cartilage in the human bodies. The purple color, this is elastic cartilage. Ordinary, this is auricle of the external ear. Also, we have talking about the tip of nose. Also, elastic cartilage. Also, some elastic cartilage is present inside the bronchi, but an epiglottis. Epiglottis is also elastic cartilage, but in other cartilage dominated hyaline cartilage, present here as you have seen everywhere. Hyaline cartilage and fibrocartilage is present in the intervertebral disc, in the intervertebral disc and in the pubic symphysis. This is fibrocartilage. So ordinary hyaline cartilage characterizes the very numerous chondrocytes in the isogenic groups. It's not a 2, sometimes it's a 4, 6, 8, and we have the very progressive increasing of diameter of chondrocytes in isogenic group between the young cartilage, juvenile cartilage, and maturated and mature cartilage between the perichondrium surround that cartilage. Well, and we have seen the hyaline cartilage characteristic. The term of derived from the Greek hyalos, glassy, because it looks like a white glass. In the most common cartilage in the body, it's located in the nose, larynx, trachea, and bronchi. It forms the cartilaginous parts of the ribs and covers the articular surface of bones. Hyaline cartilage builds also the primary skeleton of the fetus. It's a surrounding for the exception of articular surface by the perichondrium. It's a composed of the outer fibrous layer and the inner cellular layer of perichondrium. The outer fibrous layer It's a dense connective tissue that contains a collagen and elastic fibers, fibroblast, and blood vessels. The inner cellular layer of perichondrium contains the stem chondrogenic cells, chondroblast. The perichondrium provides the cartilage with the nutrients and take part of the cartilage peripheral growth and regeneration. So we have seen the types of cartilage here. This is originally upper part of photo, this perichondrium with a good developed fibrous layer and cellular layer. Also, we have seen the major cartilage and some cartilage in lacuna and special differentiation of cartilage during the maturation. This is a classical microscopic slides from Ross book. Okay, chondrogenic cells differentiate into the chondroblast from chondroblast. They compose under the perichondrium at the periphery of cartilage, the zone of young cartilage. Chondroblasts secrete extracellular matrix capable of mitotic division. They convert to chondrocytes, which are the major cartilaginous cells. They are located deeper and building the zone of major cartilage. Chondrocytes still proliferate and retain the secretory activity. They are spherical and arranged in groups of 4 or 8 cells called isogenous groups. They are appeared as a result of mitotic divisions of the initial chondrocytes and cannot be dispersed in the dense matrix. So elastic cartilage is another second type of cartilage. It's found in the ear, the auditory tube, the epiglottis, and the parts of the larynx. The structure of elastic cartilage is identical to the hyaline cartilage, but it contains elastic fibers instead of collagen, which provides. And with the yellowish color and elasticity, elastic cartilage possesses the perichondrium with the stem chondrogenic cells, as well as before. We're talking about in the hyaline. Under the perichondrium, there are the zone of a young cartilage containing the chondrogenic cells too. In the center of this zone of major cartilage, it's chondrocytes for the isogenic groups. Elastic cartilage does not degenerate as readily as hyaline cartilage, and the matrix of elastic cartilage never undergoes calcification. But what about calcification? Ordinarily, the fibrocartilage and hyaline cartilage can be calcified, and that's a result of accumulation inside the matrix of cartilage, the salt of hydroxyapatite with a time of senescent always. Okay, we have compared two types of cartilage. This is elastic cartilage and fibrocartilage. As you have seen here, that's in elastic cartilage we have the group of isogenic group, which is to form like a special range. But in the fibrocartilage, as the cells also resemble on range, but it's not to form a lot of cells in the isogenic group. Ordinary 1 and 2, and they have to elongate along the special fibers. And in elastic cartilage, dominated elastic fibers, but in the fibrocartilage, dominated collagen type 1 and collagen type 2, but doesn't present collagen, elastic fibers, doesn't present. Fibrocartilage, it exists in the invertebral disc, the pubic symphysis, and the main meniscus of the knee joint, at the junctions between the large tendons and the articular cartilage in the large joint. It serves the transitional between the hyaline cartilage and the dense regular connective tissue. So features: the fibrocartilage extracellular matrix contains the glycosaminoglycans and more collagen fibers than the hyaline cartilage. Collagen fibers are arranged in a parallel bundles like a tendon. The fibrocartilage chondrocytes lie longitudinally disposed in columns between the cartilage collagen bundles. The osteogenesis, just we have talked about the development of bone. And we first of all started for prenatal and postnatal osteogenesis. Bone formation starts in the first months of prenatal development. They continued till 25 years of life. Intramembranous bone formation to develop from mesenchyme, and more important for the reticular fibrous woven bone for development. And the chordal bone formation, this is development from the cartilage. As you have seen here, this is a mandible of the pig embryo, and we have seen here the two types of cartilage. This is Meckel cartilage is named. This is a single area where the cartilage is present in the mandible. So all another bone in mandible as well as in skull, it's originate from the mesenchyme. This is intramembranous bone formation. Look at this one. We have seen pink color area. This is named osteoid matrix of bone tissue. At the periphery, very good seen columnar cells and cuboidal cells. This is osteoblast cells. Also, we have seen the cells inside, which is embedded inside the osteoid. This is osteocyte cells. At the near it, here for example, and here, sometimes is visible osteoclast cells, but in this photo is not properly seen. But at surrounding tissue, we have seen the blood vessels, and also we have seen this is mesenchymic cells. This is mesenchyme. Okay, so osteogenesis of long bones to regulate by the endochordal ossification. Here is present all stages of this ossification, but we just to discuss the endochordal ossification and perichordal ossification during our practical lecture today. We have seen also special area of metaphysis, the metaepiphyseal plate between diaphysis and epiphysis. This is epiphysis, this is diaphysis. So, and this is metaphysis, the special area where is the cartilage to transform into the mature bone during the morphogenesis. So bone histogenesis. The bone is derived from embryonic mesenchyme. It develops in the one or two ways. Intramembranous, the development of bone directly from mesenchyme, and endochordal occurs via the cartilage model that is replaced by the bone. Bone remodeling continues throughout the life. Intramembranous bone formation, also known as direct osteogenesis, begins when the mesenchymal cells condense to form osteogenic islets, differentiate to osteoblast, and begins to secrete the matrix. On the bone surface, osteoblast can compose of simple layer of cuboidal cells. The osteoblasts are connected by their processes embedded in their oven matrix and become osteocytes. This transformation, of course. The osteoblast and bone matrix with the enwrapped osteocytes constitute bone trabecula. Bone trabecula calcify and join together. This is calcification, also a result of accumulation of hydroxyapatite. Hydroxyapatite, it's a hydrophosphatic salt. And the primary reticular fibrous bone forms the first and later replaced by the secondary lamellar bone. So it's very important. So primary is reticular fibrous bones, and after that to replace by lamellar bones. So what about intramembranous ossification can be seen here? Look at this one, the first formation of osteogenic islets. This is osteogenic islets to start from the patterning signals. We will discuss this in bone morphogenic proteins, transforming growth factor under the Hox genes, and we have seen how the mesenchymal cells aggregates near the blood vessels. As next transformation, it starts the formation of osteogenic islets. At the periphery of this is osteogenic islets is visible special cuboidal cells. This is osteoblast, and they are produced bone matrix, which is named osteoid, and the cells migrated from the osteoblast into the center and start osteocytes. The next continuous development will be calcification of osteoids. During the calcification of osteoids, that's osteocytes to join each to other through the special canals which to form the lacunar canalicular system. Each osteocytes in lacuna and the canals, this is the processes of this is osteocytes. At the periphery, the shape of bony plates to regulate by the osteoclast, specialized cell destructor, which is a modification of macrophages. This is a macrophage of bone, and this is macrophage of bone was migrated into the bony tissue during formation from the red bone marrow in the early stages around the 7-9 week of development. And the chordal bone formation. Indirect osteogenesis is typical of a long bone development. It begins in the hyaline cartilage model. Look at this one. We have seen the special schematic diagram, how the hyaline cartilage model is appear, after that in the region of periosteum to appear, after that the blood vessels migrate from the periosteum inside the cartilage and to form the primary center of ossification. After that we have the formation of trabeculae inside the diaphysis. And after, we have talking about the secondary center of ossification in the epiphysis, secondary center of ossification. And between the primary and secondary center, we have seen special, this is calcified cartilage, special area which is named metaphysis, we have discussed. And on the metaepiphyseal plate, this is area is named metaepiphyseal plate, we have the some zone. First zone, this is zone of epiphyseal cartilage, is normal cartilage, doesn't destroy, doesn't change. Next, reserve zone, where does it take place? The mitotical division, reserve zone. Next is this proliferating zone, which is very active proliferate. And also the hypertrophic zone, the zone which is present like transformation, that's increase the cytoplasm diameter of its cells. And next is a vascular invasion zone, next zone, where is this cartilage is destroyed via hypertrophic zone area and replaced by the bony plates on the bordering between the vascular invasion zone and hypertrophic zone. Okay, a primary center of ossification occurs at the diaphysis of the cartilage model of the bone. It starts from this transformation of perichondrium into the periosteum and chondrogenic cells into the osteogenic cells, which is a turn differentiate into the osteoblast, elaborate the bone matrix on the cartilage surface. The new bone appears in the bone collar or perichondral bone annulus. It disturbs the more normal feeding of cartilage from the perichondrium. Therefore, the chondrocytes within the cartilage model begins to degenerate, and cartilaginous matrix undergoes calcification. The blood vessels from the bone collar penetrate the cartilage model, and the osteogenic cells and osteoclasts come here in this area. Osteoclasts begin to resorb the mineralized cartilage matrix, and osteoblast elaborate the bone matrix formation, also known like endochordal bone formation. The bone collar becomes thicker and elongates toward the epiphysis. The osteoclast resorb the endochordal bone, establishing the primitive marrow cavity. The processes of resorb of spread toward the epiphysis where the secondary center of ossification appears. The ossification begins when the osteogenic cells from the blood and adjacent periosteum invade the epiphysis and differentiate into the osteoblast and elaborate the bone matrix to replace the disintegrating cartilage. The remaining the cartilage still available in the metaphyseal plate that will be replaced by the bone when growth ceases. And the metaphyseal plate continues to grow by adding the new cartilage at the epiphyseal end while the being replaced by the bone at the diaphysis end. At about 20 years of age, as the metaphyseal plate is replaced by the bone, the elongation of bones and the growth of the bone body stops ordinary. Okay, the bone. The bone tissue consists of its cells, osteoblast, osteocytes, and osteoclast, and the intercellular matrix, amorphous ground substance, and collagen fibers. Osteoblast. The osteoblasts are derived from the osteogenic stem cells and secreted the organic portion of the bone matrix. Therefore, they contain a well-developed rough endoplasmic reticulum and smooth endoplasmic reticulum, the Golgi complex and secretory granules. Secreting the collagen and glycosaminoglycans, osteoblasts are embedded themselves into the intercellular matrix, which will become calcified later. As a result of their cells, bodies are located in lacuna and their cytoplasmic processes in bone canaliculi. The processes of canaliculi of different osteoblasts are anastomized. Interconnected is named. Then osteoblasts stop secreting and are converted into osteocyte. Look at this one. This is a schematic picture of the osteoblast. We have seen the nucleus, we have seen rough endoplasmic reticulum, Golgi apparatus, any area of secretory vesicles, and specialized processes, collagen fibers, and we have seen the matrix formation. Next, osteocytes. The osteocytes are derived from osteoblasts. These major aged bone cells housed in the lacuna and maintain the communication with each other via gap junctions between their narrow cytoplasmic processing extending through the canaliculi. Therefore, the oxygen and nutrition can diffuse from the blood vessels within the canaliculi to nourish and maintain these cells. Since osteocytes do not secrete their bone matrix anymore, they contain condensed nuclear chromatin, heterochromatin, and reduces the amount of organelles. They maintain bone homeostasis through the canaliculi. And we have seen here the special picture of the osteoblast, or osteocytes under the optic microscope, osteocytes schematic diagram, osteocytes under the scanning electron microscopy, and classical transmissional electron micrograph, whereas we have seen the heterochromatin, as I minute ago tell you about it, at the periphery. In the center is nucleolus. Also we have seen the very good visible nuclear envelopes. Also we have seen good visible some rough endoplasmic reticulum elements is here visible. Also visible special vacuoles which produce the extracellular matrix. And we have seen the lacuna surface canaliculi, and calcified area of bony plates. Okay, next, osteoclast. Osteoclasts are multinucleated, up of 15 nuclei, and a huge amount of cytoplasm in called symplast. Why? Because a lot of nuclei in one cytoplasm. They are derived from the blood monocytes, originally they have to migrate from the red bone marrow and responsible for bone resorption. The functions of osteoclast is a destruction of bone by creating of acidic environment that decalcifies and surface bone layer. Their lysosomes release the enzymes that digest the organic component of bone and inorganic component calcium salts and dilated by the carbonic acid secreted by osteoclast. Osteoclast's composition and structure, we have seen the bone. This is bone components, and this is osteoclast. 1, 2, 3, 4, 4 nuclei, and we have the resorptive zone. Resorptive zone is visible here. We have seen the ordinary mesenchymic cells here. So this is the picture from the intramembranous bone formation. Also, we have this is a schematic diagram which is shown osteoclast structure, but sorry, in Russian language. We have seen the blood capillary. This is osteoclast. This is a nuclei. This is a Golgi complex, lysosomes. This is process of formation of carbonic acid, and this is carbonic acid to result of smooth endoplasmic reticulum. Smooth endoplasmic reticulum generates a numerous of hydrocarbonic acid, which is migrated between the specialized intracellular canaliculi, and this is canaliculi is open into the resorptive zone. Resorptive zone, this is a special area. And we have the destruction of the bone here under the hydrocarbonic acid. Look at this one. This is a photo of active osteoclast with multinucleated. We have seen also the mature bone. Alcyan blue is also used for this color staining. And we have the bone matrix contains organic and inorganic components. The organic part is composed of type 1 collagen and amorphous ground substance osteomucoid. The inorganic portion represents about 70% of the dry weight of the bone. It's composed of mainly of calcium and phosphatous salts. The salts is amorphous form of the hydroxyapatite crystals. Both bone matrix components work synergically to produce the extraordinary strength and flexibility of bone. A special construction. Since the mineralized bone matrix preserve the free diffusion in contrast of cartilage. Bone contains the blood vessels. It's very important. Maybe you know from the anatomy Folkman's vessels and of course Gaversian system of the vessels. And the reticular fibrous woven bone matrix contains the thick bundles of collagen fibers, which may lie in the 3 directions with the osteocytes between them. This type of bone occurs mostly in embryogenesis. Lamellar bone. The lamellar bone tissue replaces the primary bone. Its collagen fibers lay inside any lamella in the parallel direction. The osteocytes in lacuna are located between the lamella and occasionally within them. It is in turn the lamellar bone tissue form the 2 types of bone substance, spongy and compact. Reticulofibrous woven bone. Look at this one. We have seen the structure. This is bios particle which is embedded inside after the injuria, and we have the surround of this is particles, bios particles. We have the connective tissue around bios particles and formation of mineralized bone. This is osteoid bone formation near the injuria. This is effect of reticulofibrous woven bone, ordinary regenerative bone tissue for regenerate injuria area. Sometimes it's appear near the tendon of junctions between muscle and tendon, yes. And we have to identify this is area on the surface of the longitudinal bone near the muscle. Also, lamellar bone tissue, compact bone. This is classical special stain for compact bone identification. We have special Schmorl stain, which is contain picrofuchsin and thionin. Picrofuchsin to give rise to formation of green color, but the thionin brown and brown color. But we have seen here Haversian system. This is osteon. This is intercollected bone lamella, and we just seen another elements like here. Here we have seen the periosteum. That's very good, stained by thionin. It's have good stain, yes. And we have seen here the circumferential bony plates. Circumferential bony plates is very good visible here. This is bony lacuna of osteocytes, and lacunar canaliculus system is very good visible, and this is a cavity of Haversian system. Also, in the inner surface of bone, we have seen endosteum, if periosteum outside, endosteum inside, and we have red bone marrow with a special cells megakaryocyte of bone marrow. Well, bone as an organ. The bone as an organ includes bone tissue, periosteum, endosteum, blood vessels, articular cartilage, and bone marrow inside. So we have seen the main unit of the bone, which is named osteon. Osteon, it's a system of bony lamellas, which is embedded each to other to form like a cylindrical system, which to contain Haversian canal, and this is a system of osteon. It's a classical for compact bone, but for spongy bone, we have talking about the trabeculae of spongy bone, and the bone marrow is located in the spongy bone, in the epiphysis, but in the diaphysis ordinarily present compact bone. Osteon, structural unit. In the diaphysis of long bones, lamellas exhibit a special arrangement. There are the outer circumferential lamella and inner circumferential lamella, as well as a thick osteonic layer between them, which consists of typical osteons and interstitial lamellas between osteons. The osteon is a cylindrical shape and composed of 120 concentric lamella surrounding the osteogenic canal, central Haversian canal that contains blood vessels, nerves, and some loose connective tissue with osteoblast and osteoclast. Interstitial lamella are the remnant of old destroyed osteons. And we have seen the lacunar canaliculus system in a higher magnification here. This is interstitial lamella. Well, also regulation of exchange of calcium and phosphate in the bone to regulate by the special hormones calcitonin and parad hormone, parathyrin. That's a homeostasis of blood calcium levels is regulated by the special glands. The calcitonin is produced by thyroid glands, seed cells. And this is calcitonin to activate the osteoblast cells, but the parad hormone, parathyrin, to stimulate the osteoclast cells for destroy of bony lamella and to release a new portion of the calcium ions stimulus. But so interactions between calcium and phosphatic levels too is composed regulates by this is thyroid and parathyroid hormone, and of course the all osteoblast and osteoclast cells is controlling by, especially osteoblast cells, controlling by the somatotrophin of hormone of pituitary gland. Somatotrophin, also communicates with the levels of steroids hormones with age. Like example, famous effect of destruction of bone via activities of osteoclast under the decreasing of level of estrogens and increasing of level of androgens in female body, like ordinary's name of some diseases like process of age changes, osteomyelitis, and process of destruction of the bone tissue. Thank you very much. The lecture is finished. I wish you be healthy and good study. Goodbye, students.